I have the privilege to introduce Dr. Tony Blau. In terms of his education, Dr. Blau started in Wright State and then got his medical degree in Ohio State University before going to Duke for internal medicine residency. Around 1989, he finally came to the University of Washington for the oncology fellowship and stayed here since, kind of accelerating through our ranks until he became staff in 1994 and full professor in 2005. In 2006, he became the co-director for the Institute of Stem Cell and Regenerative Research, and in 2013, he became the director of the Center for Cancer Innovation. He's also the founder and president and CSO of Partners in Personal Oncology. He's won many awards, including the Medical Staff Teamwork Leadership and Caring Award. He's a member on the editorial board for a number of respected journals, a reviewer for the Gates Foundation. He's serving currently on many active grants, has 2 patents published in journals like Blood, NEJM, Nature, which I've heard of before. And when I was asking Dr. Bremner for some advice on introducing somebody for whom I have so respect, he said so much respect, he said to say what Dr. Blau's mother would like to hear about him. And so just very briefly about that, Dr. Blau is loved and respected by the residents of our institution. During the 4 weeks he attends on our oncology service, everyone constantly talks about his caring, his relationship with the patients, and his dedication to teaching. So, with a great deal of respect, I'd like to introduce Dr. Tony Blau.
Thank you, Cameron. Thanks very much. It's terrific to be here. It's a real privilege to be able to present here. So I'm going to give you kind of the guts of my talk in the next 2 sentences. And the first is that I believe that we're at a historic inflection point in our ability to effectively treat cancer. I think we'll look back at this time as being really an incredibly exciting time, and we're really just at the very beginning of this. So I think the potential for making dramatic advances in cancer is unlike any other time in the past, and it's greater than it will ever be in the future. I think it's incredibly exciting. The second thing is to realize that potential. Everything is going to have to change. Everything about the way things are now will have to change. Now, this first slide just reminds me mainly that I came to Seattle a long time ago. I don't look like this anymore. And I came to Seattle because I came to do bone marrow transplantation. And the thing about bone marrow transplantation that's so incredibly appealing is it's all or none. You're kind of completely in. Every patient, you're throwing the kitchen sink at them, pushing them to the brink of death with very high doses of chemotherapy and radiation. That's how we used to do it. And then rescuing them with somebody else's bone marrow. And so this win or lose mentality. So much of oncology is a compromise. Is a medicine going to cause more harm than good? In bone marrow transplantation, you don't even think about that stuff. You're kind of totally head over heels, hair on fire, doing whatever it takes to try to get a patient through a tight spot in the hopes that they'll live to be a little old man or woman someday. And this is a picture that was taken when I was on the transplant ward. This is now when I was an early attending, taken actually by a patient that we were taking care of then. And this is on the occasion of her daughter's 5th birthday. And this patient had a disease that here everyone will know called chronic myelogenous leukemia. And she sadly died not long after this picture was taken from a complication of bone marrow transplantation called graft versus host disease. And although we didn't know it at the time, we were really in the dark ages for treating CML. And only a few years later, Gleevec was available. And had this woman developed CML just 5 years later, she'd be able to probably be alive today and see her daughter today. So the idea of urgency in cancer is an enormously impressive point to me. It's impressed me my whole life. So in the in the very beginning of my career, I initially did my fellowship, then I went to the lab. And I've done research in a variety of different areas, and in moving from one area to the next, with, perhaps the only unifying theme, perhaps it's clear to me only, because it's not so obvious why you would move from one area to the next. That's not the traditional trajectory of somebody that's in academia. You usually go very deep in one thing. You become the world expert in that. And I've kind of moved from one thing to the next, with the sole idea being to try to do something that could potentially help a patient someday, and ideally within my lifetime. As I spent most of my time in the lab, my wife, Sibel, is a hardcore oncologist in private practice down in Puyallup, seeing 40 patients a day some days, really in the trenches. And although my research previously had been in gene therapy and stem cells and globin-related things, I started to go with Sibel to some of the oncology meetings about 5 years ago. And it struck me then, in going to some of these meetings, that the way we were trying to address cancer as a community was wrong, and that we needed to have a fundamentally different way of thinking about our approach to cancer. And really, I think the grand challenge is reflected here on a graph. I guess I can just point with my computer pointer here. So here we have exponential growth. These are just different growth curves, and linear growth. And and one thing that you become impressed with in the world of research is that if you plot the rate at which technology is improving, it's improving exponentially. It's breathtaking how fast technology is improving. Every few weeks you see something new, really impressive. But if you see the rate at which those technological advances are converted to the care of a cancer patient today, and how much do patients benefit from this technological progress, that rate of improvement is linear. And so if we were to place ourselves in the position of a patient with advanced cancer who's fighting for his or her life, we would be facing an ever-widening gap between what's possible technologically and what's brought to bear in our care. And I think collectively, the grand challenge of our time is to try to find a way to close that gap. One of the biggest technological advances everyone here will be aware of, is that the cost of DNA sequencing has plummeted. And that's this is a graph that probably many people will have seen before, but the $1 million of sequencing in 2000 costs you about a nickel today. And so it's this incredible improvement, an incredible reduction in costs. It makes it possible to think about doing DNA sequencing in ways that we could never even imagine before. And so the fundamental lesson of all of the sequencing that's been done with respect to cancer, and now thousands of different cancer specimens have been sequenced, is that every cancer is different. So let me say that one more time. Every cancer that has ever existed is different from any other cancer that has ever existed or ever will exist. And I think that our field is only beginning to come to terms with the profound implications of cancer's uniqueness. Now, why is this fact important? Why is cancer's uniqueness important? And I think it comes down to how we think about advancing biomedical research. Everyone in the room will know that the primary evidence upon which we base our decisions in medicine is related to the randomized clinical trial. And so we don't believe anything if it hasn't been proven in a double-blind, placebo-controlled randomized trial. And let's say in breast cancer for a moment. Let's just say in breast cancer. Let's have a patient with breast cancer. We want to figure out how to improve the forefront of treatment in breast cancer. So you find one breast cancer patient, and you find other patients that kind of look like her, and then you randomly assign them to two different groups. Pop them into different boxes, and then you treat one group with a standard treatment, the other group with an experimental treatment, and essentially you see who does better, which group does better, and that's shown here with the boxes. So let's imagine that the women here that have the boxes survive, the other women don't, and we move forward, then we just see, okay, which treatment produced a better result on average among a group of patients compared to the other treatment. And in this case, it's a standard treatment, so we move forward with that, and we cast aside the experimental treatment. Now, this approach might work if we thought for breast cancer that maybe there are, just a limited number of subgroups of breast cancer. How many kinds of breast cancer do you think there are? Imagine for a moment that it's like when you go to the grocery store. And let's say, how many fruits and vegetables are there in the produce department? And maybe there are 50. It's a guess. I haven't actually counted. But maybe if there are 50 different types of breast cancer, and you grab, thousands and thousands of patients, and you randomly assign them between different groups, maybe those 50 different subgroups will kind of even out between the treatment group and the control group. But what if the number of different types of breast cancer is really more like every single item you could buy in Manhattan? What if there are that many kinds of breast cancer? There is no number of patients that you can enroll in a randomized double-blind placebo-controlled trial that will account for that type of heterogeneity. And as I told you already, this is what breast cancer is like. Breast cancer is even more unique than this. So, and just to go with to say one more thing about the randomized trial, we could imagine, let's imagine that we're in that trial, or one of our family members is in that trial. We could say, gosh, I hope that I get in the trial that works better for that's demonstrated to be superior. But there will be people who got that treatment, the superior treatment overall, that did not respond, who would have responded if they had been assigned to the other subgroup. And we have no way of capturing that type of information. So toward that end, after thinking about this for a long time and working with many other people that I'll describe as the talk goes along, we thought that there needs to be a different approach to trying to tackle cancer. And so we made something here at UW-Medicine called the Center for Cancer Innovation. And that's centered around several big ideas. The first idea is that if you really want to tackle cancer, you have to try to understand what's going on in a patient with cancer. You can't treat that patient like a black box, like, oh, it's impossibly hard to figure out what's going on. You have to first believe that it might be someday really possible to understand what's going on, and that we might be able to intervene based on that understanding. So first you have to kind of believe it's possible. And then the second thing that's not hard to believe is that in order to really address this incredibly difficult problem, you need people from many different areas of expertise who work together. And I think everybody talks about this, right? Collaboration is important. We should all work together. We should link arms, sing around the fireplace. But the way that we try to aggregate the expertise of people from divergent areas of research is to focus their efforts not on cancer as a concept, but on the problems that exist within an individual patient with cancer. The other idea is that this is such an incredibly hard problem that you can't be restricted to working with people at your own institution. You have to find a way to work with anyone in the world that you want to work with. In other words, you have to find a way to transcend institutional boundaries. Anyone with the advice, technology, expertise, and most importantly, temperament, that they're willing to throw some time at this and to join forces. They drank the Kool-Aid. They kind of believe that this is a worthwhile idea. So you have to find a way to work with anyone in the world that you want to work with. The other idea is to think of cancer fundamentally as an information problem, a big data problem. If we can sequence the DNA and the RNA, if we can essentially think of a cancer as being organized in a way that's unique to the patient, but that is using rules that are standard across all of biology, then we can think of that by creating this data set, essentially digitizing a cancer patient and their cancer. What it ends up being is it ends up being a billion piece erector set. And I know that many of the people here under 30 won't know what an erector set is, but it was this cool thing, believe me, where you would get you could build these little cars or houses or whatever you wanted from screws and nuts and bolts. But this is a billion-piece erector set that has no instruction manual, or has the barest of instruction manuals. So thinking of cancer as a big data problem. And if we think about it in that way, then you could think of people who have expertise in big data as being really important in trying to tackle this cancer problem. I mean, how much effort is put into trying to figure out whether the Dow Jones Industrial Average will take a tick, of a fraction of a percent up or down? How much effort's been put into that? And what if you were to begin to think of placing that much effort in service to an individual patient with cancer? And then the other idea is to fuse research and clinical care. So typically, the way we think about things is in two spheres. There's the sphere of research and the sphere of clinical care. And we have pretty tight safeguards that separate these two spheres because we don't want our patients to be subject to harebrained research ideas that actually could harm them without offering them any benefit. But we believe that in patients with advanced cancer, that this barrier can prevent patients from accessing some of the very best and latest technologies that the world has to offer. And so in the types of studies that are supported by the Center for Cancer Innovation, we remove that barrier and we fuse research and clinical care. And so as an embodiment of this type of idea, we've had many meetings with many different people. One of the big initial meetings was a meeting at our house where, Isabel and my house, where we had all of these people show up. And the interesting thing here is that they represent many different areas of scientific expertise. We have computational biologists, hematologists, cell biologists, hardcore researchers. We have academic oncologists and hematologists. You'll notice Mike there in the background. We have fellows, trainees. We even have a high school kid who wants to be a doctor someday. So the whole thing, find anyone who wants to work on this. But the other thing you need is you need patients who are willing to be the subjects of an unprecedented scientific investigation. And in this picture is Misa Ingalls, who has, and she's right here, who had a type of breast cancer that's the subject of our initial clinical trial called metastatic triple-negative breast cancer. And Misa, sadly, this picture was taken last November. Misa sadly died earlier this year. The way that our clinical trial, the idea for the type of clinical trial that we're doing is that since every cancer patient is unique, you can't go study a mouse with a breast cancer and necessarily learn rules that will be relevant to the patient that's sitting in front of you right now. It's such a complex ecosystem, that patient, where they have a tumor that's uniquely compromised, composed of, and driven by a complement of mutations that has never occurred in any other patient. It's surrounded by a response from the microenvironment and immune system that's specific to that patient. And so there is no in vitro or lab-based model that will mimic in any way that comes even close what's happening in that patient. So in the clinical trials that we support, we find patients who face this incredibly difficult problem and who want to participate in a clinical trial where we tell them that they're very unlikely to benefit directly from their participation, because despite our very best efforts, we still don't know nearly enough to help the vast majority of our patients. We sample the tumor at many different sites of their body. We biopsy it at many different sites. The reason for that is that the cancer is not only unique to every individual, but within a patient, it's under Darwinian selection. It's constantly evolving, and you cannot assume that cancer at one site in the body is identical to cancer at another site in the body. In fact, it's not. And Larry Loeb has proven this, better than anyone. So we are trying to encompass that heterogeneity that exists within an individual cancer patient by doing multiple biopsies. We place this information on the cloud, so we digitize a patient, we collect billions of data points, and we place it on the cloud, and we make it accessible to some of the best computational biologists in the world who look at it, try to place this information in the context of prior knowledge, and the initial effort is all about, can we find something in this tumor that might point to a vulnerability that could be attacked with a targeted drug? And so we do our best. We take our best shot. And we convene a tumor board, and we develop a prediction, a hypothesis, if you will, about what's going on in that patient. And then we test that hypothesis, not in a petri dish, not in a lab animal, but in the patient, by figuring out a way to get the patient on the drug that's predicted to work. And then you use what happens in that patient. Did the tumor respond or not? And feed that information back to the computational people, creating a learning loop, closing a learning loop. You can think of this as something like efficient hypothesis testing, one patient at a time. And when our predictions are wrong, and we know that they'll be wrong frequently, we repeat a set of biopsies and we take our next best shot. And through this process of what we call intensive longitudinal monitoring, with multiple biopsies at multiple time points over the course of the patient's illness, before and after treatments that are designed to help them based on our best available understanding at any given point in time, what we're really aiming to assemble as we do this in tens, hundreds, and eventually thousands of patients is to assemble a database that we believe will eventually amount to an instruction manual for how cancer works. When I was an intern at Duke in the 1980s, HIV was rampant. There was no treatment. You got HIV, you died. And then during the internship first part of residency, AZT came along. And the virus would go away, but then it would come back, having developed mutations that would allow it to resist treatment. And then we gave the next drug, and the virus would go away, and it would come back. And over the decades, we've learned the rules of how HIV evolves and escapes treatment. And today, as everyone here knows, when you get HIV, you get a cocktail of drugs that not only block the virus now, but also block off the anticipated mechanisms of escape. We know what the rules are for HIV, and today HIV is a chronic disease. We don't know what the rules are for cancer. It's a massively more difficult problem. But I think we need to begin to build this instruction manual that will teach us how cancer works. So the clinical trials that we're interested in at the Center for Cancer Innovation are different from standard clinical trials in a number of ways. The first is that, as I've already said, standard clinical trials have to, randomized, double-blind, placebo-controlled trials have to assume broad similarities between different patients with the same disease. They're looking for differences in average differences between groups of patients. And the trials that we're developing assume they actually embrace the idea that every cancer is unique. A standard clinical trial is very much focused on testing a treatment. So you get a treatment of interest, say, some new thing that we want to test, compare it to standard treatment. We take that new treatment and we run hundreds, dozens at least, hundreds, sometimes thousands sometimes of patients through that treatment. And so it's all about testing the treatment and running lots of patients through that treatment and seeing whether it has a benefit. The clinical trials that we're pushing forward at the Center for Cancer Innovation doesn't care what the treatment is. We want to find, if we have a bead on a treatment that we think might be helpful to our patients, that's the one we want. As I've said already, third point I've already said already. Fourth point is that standard clinical trials require large groups of patients upon which to base scientific advances. So every nugget of knowledge comes from what happened in typically hundreds or thousands of patients. So quanta of learning go down to hundreds or thousands of patients. What we're aiming for, and this is really controversial, but is it possible that the quanta for learning could be an individual patient? Could we potentially learn just a smidge, just a little bit, from each one of the patients that go through our clinical trial? And is it possible, by doing this iteratively, that we eventually uncover some generalizable knowledge? How do we convert what happens in a single patient from an anecdote to real knowledge, real science? How do we make this real science? The initial clinical trial that we are doing, as I've already mentioned, is in metastatic triple-negative breast cancer for people that aren't aficionados. That means breast cancer that lacks HER2, which is a major driver of some breast cancers and can be targeted with a treatment. It also lacks estrogen receptor and progesterone receptor. So there are, which can also be targeted. So there are no targeted therapies for triple-negative breast cancer, and metastatic triple-negative breast cancer is an incredibly aggressive disease. There are reasons, I think really good reasons, why we decided to focus on this, but it's a very tough problem. So, so to enroll in our study, the patient has to be scheduled to receive a drug called cisplatin. And so our protocol is not about treatment. We're not really involved at all with the treatment of the patient, but in order to qualify for enrollment in our protocol, you and your oncologist have to have decided that the next treatment you're going to get is cisplatin. And really that does two things. That brings everyone into a common initial path of treatment, so that even though these patients will have received many different treatments in the past, in this study, they're all getting treated in the same way in the first initial step of this study. The second thing it does is it buys us time. That patients aren't waiting around for us to get our act together before they actually start treatment. And we found uniformly patients cannot afford the time to wait. You have to start something right away. So again, the way this trial is designed is in these patients, we do multiple biopsies. That's reflected by these bars here, biopsying multiple different sites of disease. We do a number of different molecular assays that I'll talk about in a moment. We place the information on the cloud. It's analyzed by a number of groups, and really big contributors are from the laboratory of a guy named David Hausler, who's like a world expert in cancer genomics and in computational biology, but many different institutions are looking at this. Colin Pritchard in lab medicine and the Oncoplex assay have been immensely important, and Mike Dorschner here in precision medicine initiative has been immensely important. So as this information is sitting here, we're looking at it, trying to integrate it, and we have a meeting of what we call our atomic tumor boards. We call it atomic because we call it the intensive trial of omics in cancer. It's always an enormous challenge. You have all of this information, all of these mutations that are happening in the patient's tumor, trying to figure out which ones are important and trying to target those is a huge challenge. We create a report, and the idea is to have a recommendation for a treatment that's available by the time the patient needs it. And that means by the time that they're finished with cisplatin. And they can be finished with cisplatin either because the tumor is growing through cisplatin or because cisplatin is causing toxicity and they just can't take it anymore. And so at the end of cisplatin, then we again do multiple biopsies, and then we work very hard to get if the patient and the oncologist want, we work very hard to try to help them get access to whatever treatment's recommended. This is just a schematic showing some of the information about the different tests. The whole exome sequencing sequences approximately 20,000 genes. We're comparing there the genome of the patient's normal cells that we get from their blood with the genome of their tumor cells, and again, from multiple different representations of their tumor. The Oncoplex assay was an assay that was developed here by Colin Pritchard and his colleagues in lab medicine, and it sequences 194 cancer-associated genes. You could think that's redundant. Why are we doing 20,000 on one hand, 194 on the other? But there are good reasons for this, and it's been super helpful. And then we do RNA sequencing, so we look at which genes are turned on or off. And so I'll just tell you about a few of our patients. Our first patient was a 45-year-old woman with metastatic triple-negative breast cancer, really bad, really aggressive, growing through lots of prior treatments. This is a CT of the chest showing a right axillary lymph node that was biopsied 7 times in the ultrasound suite, so core needle biopsies. We also did core needle biopsies of a left chest wall mass. And we have this really elaborate, we spend an enormous amount of attention on assuring the integrity of the tissues that we get. And doing, this is just kind of showing it's complicated. We've put a lot of thought into how these tissues are processed and how they're collected, because what can happen is if you're not careful, artifacts can enter into the specimen that can really mess up all the downstream processing. So we spend a lot of time with this. We work very closely with Steve Schmeckel and his colleagues at Northwest BioTrust. And when we're in the when we're in the ultrasound suite, sadly the autopsy suite comes up at some point, but when we're in the ultrasound suite, you can see the people so here's the patient. These are the people doing the ultrasound and the biopsy. But back here are all the people that are handling the tissue, making sure that so the pathologist is available immediately to say, okay, are we in tumor or not? What do we think the tumor content is? Do we have to do another biopsy? We take pictures, we measure time frames. So it's a big deal. And it's worked, and we've learned a lot. I should say we've now enrolled 7 patients. We've learned a lot. We're much better at this than we were at the very beginning. This first patient, she had many, many changes. One of the changes that she had in her tumor was loss of BRCA1, this gene that Mary Claire King discovered, for which she won the Lasker Award just this past week. This patient did not have a familial BRCA1 mutation, so nothing in the germline or normal genome. But it looked like her tumor completely lacked BRCA1. And so that led to, we got our tumor board together. We looked at all of this. Liz Swisher, who's been with Mary Claire for many years and is a faculty member here, thought that a PARP inhibitor, a class of investigational drugs, might be just the ticket for this patient. She had responded to cisplatin. Her lymph nodes shrank. She had pleural effusions. Those went away on cisplatin, but she couldn't take cisplatin anymore because of toxicity. And so the decision was, let's stop cisplatin. And this is a decision not made by me. It was made by her oncologist. Let's stop the cisplatin and let's get her on a PARP inhibitor. So let's look for a clinical trial of PARP inhibitors, and there are none. So to get this patient on, because they're not approved, you have to get them as investigational agents. So to get this patient on a PARP inhibitor, we made a clinical trial only for this patient, this so-called single patient IND. She got the PARP inhibitor, she stayed on it for 2 months, and her tumor came rushing back. So there's a prediction, and it was tested, and it was wrong. Her tumor came back. We rebiopsied the tumor. Tumor actually came back in the skin. We rebiopsied the tumor, and we were interested in this other mutation that her tumor had, and that involves a gene called FGFR2, fibroblast growth factor receptor 2. And this gene had 2 point mutations, so-called point mutations, amino acid substitutions, that had been previously associated with cancer, but never before had been seen in breast cancer. So this S252W mutation has been seen in 44 women with endometrial cancer. The Y375C mutation has been seen in 6 other patients with cancer, but never breast cancer. And there's one patient with endometrial cancer who had both of these mutations, exactly both of these mutations. The other thing that you can do when you get all of this information together is you can compare across platforms. We're looking at this mutation at the DNA level, but we're also looking at it at the RNA level, the expression level. And what you find is, in this patient, it turns out to be kind of an interesting feature, is that if you look in the DNA, about half of the times that you look at that sequence of the DNA, it's mutated, which means probably that in the cancer cells, 1 copy of that gene's mutated. And so it's about a 50-50 ratio of the mutated and normal part of that gene. But if you look at the RNA, 80% of the RNAs has that mutation. And that's suggesting that there's some selective pressure within the tumor favoring expression of the mutant allele. As it turns out, that we could not get this patient on an investigational FGFR2 inhibitor, but there's an approved FGFR inhibitor by a company named Ariad. It's not approved for breast cancer, and if your drug's not approved for breast cancer, insurance won't pay. And insurance didn't pay for this, and it's kind of a relevant point because these drugs cost about $10,000 a month. And so, but in contacting the drug company, they provided the drug for free. It's called ponatinib. And so if you look at the patient's skin, these might be hard to see from, but the patient started ponatinib on May 9th. I think the easiest picture to see is here, where she had diffuse involvement of her skin. This is just 3 days after starting ponatinib, but with successive pictures, her skin cleared. And so not complete, and then about 2 weeks after this last picture was taken, the patient had a small bowel infarct and died. And we don't understand exactly what that was due to. It's possible that it was a complication of the ponatinib, which can cause arterial thrombosis, although at autopsy we didn't see that. At autopsy, what we did see is that there's still a lot of tumor there. So she didn't get a complete remission or anything close to a complete remission by this type of treatment. But, and you could think, okay, gosh, wasn't that a waste? All that effort. And at best, we got a partial remission, a partial response. But, imagine. You guys don't think about this all the time, but in CML, chronic myelogenous leukemia, we all know BCR-ABL is the thing that drives that, and if you treat, and it occurs in different phases: chronic phase, accelerated phase, blast crisis, and you can give Gleevec during blast crisis of CML. And it might cause the CML to go away for a little while or to respond a smidge, but it's going to come roaring back every time. Whereas if you gave it in chronic phase, people can live for decades. And so by the same token, had we known at the earliest time point that this patient's tumor had this complement of FGFR2 mutations, could we think at that point about intervening and potentially in that context having a treatment that could have helped her for a much, much longer period of time? Second patient is a 56-year-old woman who had breast cancer originally in 2007, treated, gone for many years, until last summer. It's actually now 2 summers ago, so summer of 2013, when she had a really low platelet count. And on a bone marrow biopsy, 90% of the cells in her bone marrow on a bone marrow biopsy were breast cancer cells. And so she got a number of different treatments. None of them worked. I met her last October. She enrolled in our trial. She had a platelet count of 21,000. The only place where we could get breast cancer cells was from her bone marrow, because a staging study showed bone disease, but no disease anywhere else. And so this was actually the first patient that we biopsied on our protocol. And I never thought that I would be the guy to do our biopsies, but I'm a hematologist, and it's the one thing I know how to do, are bone marrow biopsies. So we have these kind of crappy cell phone pictures of the different, I did 5 bone marrow biopsies in total, 3 from the left iliac crest, 2 from the right iliac crest, and dissociated the breast cancer cells. We brought them up to Brent Wood and Katie Doherty in hematopathology at the SCCA. Katie spent all afternoon chopping these up, shaking out the breast cancer cells, doing flow cytometry, and these red peaks just show the breast tumor cells in the different samples. And then we sequenced each different bit, so 3 different bits of tumor we sequenced. One of the things we found was, and this is work from David Hausler's group, is this is a little bit hard to see, but on the x-axis are shown some of the genes that were mutated in this patient, and then along with the frequency with which they are mutated in other patients with cancer. So this is a screenshot of something called the cancer genomics browser that David Hausler's groups created. But the idea is, let's compare what's going on in this patient with every other patient with cancer. So this is showing the frequency with which these genes are mutated in head and neck cancer, lung cancer, and breast cancer. And the only gene that's frequently mutated across all these cancers is P53. Not much you can do about that. And these other genes that are mutated in our patient are rarely mutated in other cancer patients. And so you end up with this, again, laundry list of genes. What are we going to do to try to find something that could potentially help this patient? And in this regard, I picked up on this gene called, I'll try to find, it's where the arrow is, you'll have to probably trust me, but it's a gene called ROS1. And ROS1 had a mutation then in this patient, and that's interesting because ROS1 mutations in lung cancer occur in a small fraction of patients, and when they occur, they confer susceptibility to a drug called crizotinib. If you have a ROS1 mutation in lung cancer, you have a really good chance of responding to crizotinib. So, but the kind of mutation that our patient had in ROS1 was fundamentally different from any of those lung cancer ROS1 mutations. The ROS1 mutations in lung cancer are fusion genes, where part of one gene is fused to the other part of ROS1. And this patient had a point mutation that's just a single amino acid substitution. And so could this be causing hyperactivation in ROS1 in a way that's never before been described? And so, who knows? But what you can do is you can do a PubMed search. You can find out who out there in the world is an expert on ROS1 and what do they think. So you shoot out emails to people in Taiwan, to Germany, to Tufts University. And what happens is when you say, we have this patient with this mutation, you're the world's expert, what do you think? You get this tutorial by the world's experts. They answer quickly, and they give you their best ideas about what's going on. And the guy in Germany sends this figure that's adapted from a published paper where he circles the tyrosine that's mutated in our patient. And it's in a really critical part of the protein called the kinase domain between the catalytic loop and the activation loop. And it's evolutionarily conserved. So it seems possible. But the most interesting email was one that I sent to David Baker, who's a professor of biochemistry and heads this Institute for Protein Design. David forwarded my email to his colleague Lance Stewart, unbeknownst to me. And unbeknownst to me, Lance forwarded my email to this guy, Dan Flynn, in Lawrence, Kansas. And the way Lance describes Dan Flynn is, few people on the planet know more about this than Dan Flynn. And so Dan Flynn sends an email back last February saying he thinks that he has a hypothesis about what might be going on here. And essentially, he thinks that this mutation might prevent this ROS1 from being degraded. And so he thinks that it's possible that it could be an activating mutation. In the meantime, we were measuring circulating tumor cells in this patient, and that's what's shown here in conjunction with a local biotech company called RareSite. So circulating tumor cells look low here, but they're actually really high. She started cisplatin treatment, they jumped up, but then they went to really low levels. And with the cisplatin, so her tumor was responding, her platelet count improved. And to get access to her tumor cells, so her platelet count improved, but then she couldn't continue cisplatin. She had dural involvement with her tumor here. She got radiation here. She was off cisplatin. We wanted to resample her tumor, and to resample her tumor required that we sample them from the blood because we couldn't find tumor anywhere else. So we modified our protocol to allow for leukapheresis, and Mike Lindenberger and his team kind of did all of this for us, where we could try to collect cells from the blood and try to isolate tumor cells that way. In the meantime, her doctor decided to test whether or not crizotinib could work in this patient. And crizotinib didn't work. Another hypothesis that was clearly wrong. Her circulating tumor cells shot up. In the meantime, we were looking at her leukapheresis cells, the cells that we collected from her blood. With Mike's team's help, we collected these cells. We brought this bag of cells to Shelley Heimfeld's lab at the Fred Hutchinson Cancer Research Center, where they sort out different cell populations. They were able to sort out breast cancer cells from the blood. And then we tested these breast cancer cells in a high-throughput drug screen against 180 drugs. And what we found was that of 180 drugs, the only drugs that seemed to affect the breast cancer cells both inhibited a protein called BCL2. And that's just, and we worked very hard to get her on a BCL2 inhibitor. I tried very hard. There is a BCL2 inhibitor that's in advanced clinical trials in lymphoma. And as hard as I tried to get a BCL2 inhibitor, so it's another single patient IND for this patient, the drug companies refused. She got conventional chemotherapy here, but died not long after that. And what we found was that her circulating tumor cells at this point had formed these massive aggregates, little microemboli of circulating tumor cells. And so in closing, my biggest concern about what we're doing right now for our patients is we have a lot of smart people that are involved in this. We have, our tumor boards. Anyone here would be so welcome to come to our tumor boards. If you want to come to any of our meetings, you're so welcome to come. Send me an email and we'll add you to our listserv. But we're only as smart as the people that know about what's going on in our patients. And so to try to address this problem, we've started, last week we launched a website in which we have now placed the information from one of our patients that I haven't shown you here, patient 5, and I'll just try to go to that really quickly, where anyone here can sign up, log on, agree to the terms of use, that you won't do anything evil, try to determine the patient's identity. This is patient 5 in our clinical trial. This is the number of people that have looked. And then you can look at this is a timeline of the patient here. Oh. Well, never mind. Thanks, Chuck. It looks great here. So the idea is we have all of the sequencing data. We list all of the mutations. You can vote for which mutation you think might be helpful that might be a driver for this patient. So you vote. And the mutations with the greatest number of votes rise to the top. You can vote for which treatment you think might help this patient. And again, it tallies them, they rise up. Or you can vote for which clinical trial you might think would be of help to this patient. And then there are places for comments. You can justify why you think this or that might be a good idea. And so what we're really trying to do is we're trying to crowdsource this patient's tumor, trying to really bring in the expertise of the world so that we can really do what I think it qualifies as today's definition of what it means to do our best. And with that, then, there are just many, many people to thank, and I thank you for your attention.
Could you comment on the insurance pre-approval or the Obamacare coming up? What do you cover?
So insurance pre-approval. So I think so far we haven't had any. Through fundraising, it's been possible to pay for all of the costs of the biopsies. So we're not asking insurance companies to cover any of that. What I've heard is, and I'm no expert at this, I shouldn't even be commenting on this, but that Obamacare might be good for people that are enrolled in clinical research studies, and the biggest issue that we face is if a patient suffers a complication from the biopsies, those complications, although rare, with a liver biopsy, you could have a big complication of that, and who pays for that? And so I understand that there are recently announced changes to the UW compensation plan that pays for research. I don't quite understand how they affect this, but I think that's the biggest immediate issue we face. I think kind of a broader issue is, how do we get access to these drugs? The one drug that we wanted to test, that BCL2 inhibitor, the company's going to start a clinical trial later this year in breast cancer. So why couldn't they give us a drug now? And so, and it's easy to, sound naive as an academic guy asking these things. Susan Desmond-Hellman, the new director at the Gates Foundation, has a lot of experience with this, and she thinks that we just need to have a different structure for going after these things in a way that'll be amenable to the drug companies.
That was actually my question as well, towards that conversation that you had with the drug companies. I mean, you mentioned Gleevec, and I know that was something that sat on the bench for a while because no drug company wanted to fund it. And, I mean, you're talking about an N of 1, and I don't think, Gleevec's $1 billion market share. So today, I think they were wrong, and I think they'll be wrong again. It sounds like this is amazing work. And so I was curious to hear about that. And then how do you even sell this to the drug companies? I mean, it sounds like a lot of noise, and to hear a clear signal through that with regard to data and then also what you're doing with patients must be enormously challenging. So I was just curious.
Yeah, the arrangements that we aspire to develop with drug companies go something like this. We're putting in all of this effort to try to find the patient that we think is best suited to respond to whatever drug you're testing. And no matter what happens, these patients are the most intensively monitored patients with cancer ever. And we're going to biopsy the tumor afterwards, and we're going to give you back all of that information. Now, there will be a lot of noise. There will undoubtedly be a lot of noise. But this will be a data set unlike any other data set that you'll get from larger scale clinical trials. And we're not setting out to prove anything. The fact that the FGFR2 inhibitor worked in our patient, it might not have had anything to do with FGFR2. Scientifically, we can't prove that. But the kind of thing that I'm interested in moving toward is kind of this idea. So remember the patient with the ROS1 mutation in crizotinib, and boy, did that not work. Circulating tumor cells skyrocketed. But let's imagine a different universe where it worked. Let's imagine crizotinib worked. Would that have proven that the ROS1 mutation was causal, a real driver? Of course not. But let's imagine another leap of imagination. Let's imagine that on crizotinib, she responds, but then the tumor grows back. And let's say we resequence the tumor, and let's say now the tumor has a mutation in ROS1 that prevents crizotinib from working. And now let's say we give another drug that can still work on ROS1 in that setting, and the tumor responds again. I mean, how far away from that? How far is that from real scientific evidence? And those are the kinds of things we want to work toward.
In HIV, a combination worked. Can you use a combination against any altered gene in this situation?
Yeah, I think it's going to
concentrations.
Yeah, I think it's going to be such an important question, Larry, about how to figure out how to use combinations. I think I think clearly to have any sustaining effects is going to require some way of figuring out how to use drugs in combination. I think that's super clear. I think we're just right now in our absolute infancy in this process. But that's definitely something we'd aspire to do. Chuck.
So, Tony, this is a whole new way of doing clinical work.
Oh, sorry, Dave.
Are we done?
No, David, yeah.
Okay, I'm sorry, go ahead.
So, Tony, I mean, this is really exciting. Clearly, it has to compete in the marketplace of ideas and healthcare dollars. What's the objective yardstick according to which you evaluate whether you're successful or not?
Yeah, that's a great question, David. So I think Chuck was going to ask me an easier question. So I think, it makes absolutely no sense to spend all of this money on an individual patient, with the hope of trying to get them to live a few more weeks. Makes zero sense. The concept that we're trying to push forward is this idea of all for one and one for all. All for one, bringing the world's expertise to bear on the problems of an individual cancer patient, and one for all, using what happens in that patient to teach us all about how cancer works. So the extent to which what we learn from each one of these patients can add to the generalizable knowledge that could be applied to other patients, I think that's where we hope to see value. And personally, I think that if we're going to cure cancer, this is what we have to do. So Chuck, you've got
the microphone now. So I guess my question is, what does success look like in the end with this, and how do you prove that this is a superior approach to cancer treatment compared to the traditional binning into randomized placebo-controlled clinical trials, et cetera?
Yeah, that's great. So I think the road to really, having big effects on cancer is gonna take decades, and it's gonna be hard, really hard. So I think how do we measure success in the interim? I think the first metric for success is, can we get people to work together? And that's one thing where we've been fabulously successful so far. People from all of these different organizations kind of putting their own interests, subjugating their own personal interests to trying to work toward this grand project. So I think that's already a major success. And then as we do this in more and more patients, we will end up generating hypotheses. We have one hypothesis already, or we have one little nugget of an observation already that suggests that FGFR2 might be a reasonable target in patients with cancer. So, would that be relevant if another patient with an FGFR2 mutation popped up? I'd argue that I'd want to know about this if I had another patient like that. But I think really what we want to learn are the rules. We want to learn how tumor cells evolve and escape treatment. That's going to take probably thousands of patients.
But in the end, don't you have to have outcome data that say this approach provides superior outcome to binning?
Yeah, so I think, the kinds of things that we would need to do, so let's just take the FGFR2 idea a little bit more. Let's say that we wanted to do a clinical trial in patients with breast cancer with an FGFR2 mutation. There might be a time where we would say that, let's find a handful of patients. We're going to have to, since this is the only patient that's ever been described with breast cancer and that mutation, you're going to have to get a lot of patients. Let's imagine that it's possible to get a handful of patients. And under those circumstances, you're going to probably still have to do randomization up front to test an FGFR2 inhibitor with something else to prove that this is effective. There'll have to be crossover so that everyone can eventually get the FGFR2 inhibitor. And I think, but I think the advantage of that type of incredibly focused approach is that that you need hopefully far, far fewer patients than you need today. So thanks very much.